So, good morning or good afternoon, everybody. It's my pleasure to continue our Global IMMUNOTALK series with another excellent speaker, Jonathan Kagan. And I'd like to briefly introduce John to you. John is a leader in defining the molecular and cellular pathways by which the innate immune system recognizes infections and tissue injury. He has worked at the interface of host-microbe interaction for more than 25 years, first as a trainee at Yale and since 2007 at Harvard. Here, he runs a research lab based in Boston Children's Hospital. He is the Marian N. Neutra Professor of Pediatrics at Harvard Medical School and the Director of Basic Research and Schwachman Chair in Gastroenterology at the Boston Children's Hospital. His primary research interest is to understand the molecular basis of inflammation. He has made very significant discoveries in the area, most notably related to inflammasome pathways stimulated by toll-like and RIG-I-like receptors. CGAS and STING and the inflammasome are other research focus from him. Each of these pathways are key to the induction, as we all know, of anti-infection, anti-tumor immunity, and his excellent work has helped to establish a comprehensive map of cellular sites of innate immune cell signaling and subcellular sites of innate immune cell signaling. He's also a co-founder of Corner Therapeutics, where he serves now as an advisor, and he's advisor to many or several other companies in the immunotherapy space. Jonathan's work has been recognized by awards from the American Association of Immunologists, the Boris Wellcome Fund, and the International Endotoxin and Innate Immune Society. He's also an elected fellow of the American Academy of Microbiology. And I could, of course, continue with highlighting grants and other major achievements and the many high-profile papers that you have, but I'd like to handle actually the word quickly over to you, John, but not without asking our usual question, our opening question, which is today, or my question for you today is, what do you believe is the most critical skill for a scientist to develop early in his scientific career? Thank you, John, for being here.
Terrific. Thank you, Dietmar. Thank you to all the organizers for putting together what is really a very special forum and event for the immunology community. And I want to say hello to everyone from the United States and around the world who may be logging in. So before we get started, I'll answer the question that was just posed, which is what is one of the key aspects of early career science success? And I would argue that the most determinant for me when I was coming up as a young scientist was passion for the projects that I'm working on. And the reason why I bring that up is because as we as scientists all know, there's a lot of guesswork, a lot of trial and error in the scientific process. And graduate students, postdoctoral scientists, they often can test multiple hypotheses before they finally, if they're lucky, settle on one project that actually has legs and can yield important insight into the field. That process, if you don't feel that passion for the work, can become very laborious. Yet if the passion is there, the trial and error that's needed in order to finally arrive at a clean hypothesis with testable tenets is very, very different. And so one of the main challenges that students have these days is that they may be attracted to labs that are small or large. They may be world famous or, let's say, niche famous. But it doesn't matter if you're in the biggest lab in the world. If you don't like the work, if you're not passionate for the work, you're not going to be as successful as you could be. And the opposite is also true. You could be working in a lab that is just starting out, brand new professor, for example, who may have a local reputation. But if that work that you're doing is exciting to you, that you're passionate about it, you're going to get great things coming. And so I would suggest to everyone to make sure you're focused on the people you're working with, make sure you're focused on the excitement of the project, and don't worry about where that project will go, because your famous PI doesn't know where that project will go at the beginning either. The most important thing is the work.
Well, I think that's a very excellent opening statement, and well, now I think we all want to hear more about your passion for science and what you are so passionate about. So thank you very much again for joining us. Now it's time, I think, to turn up your slides, and we look forward to the presentation.
All right.
Appreciate it.
So thank you again to everyone. We're gonna now jump into the main event, which is the discussion that I'd like to have today about how we can use the power of the innate immune system to control protective immunity. And this is primarily in the context of cancer. But as a form of background, what I'd like to do is provide the audience with a bit of context for the types of questions that my lab has typically focused on over the years, with the central one being at the top of your screen. How do we distinguish between our own cells and the microbial cells that we may encounter? This question of self, non-self discrimination has really sat at the centerpiece of the thinking of immunology as a field for many, many decades, over 100 years. And typically, what we think is that we have our own cells, which could be, for example, a macrophage or a dendritic cell that has a healthy or symbiotic relationship with other cells within our bodies that share the same genome. And that's how we define self, cells that have the same genome as ours. And we typically think that when we encounter cells that have different genomes, like bacteria, we have an antagonistic or non-productive interaction. This type of dynamic and dichotomy between healthy interactions on the left side of your slide and non-productive interactions on the right side of your slide has really led to a number of questions of how does this happen at the cellular level? How does this happen at the molecular level? But we know that this schematic is not representing all that happens within our bodies. And in fact, we know that there are bacteria that are beneficial to the host, not always problematic. And we also know that not all self cells are good cells, in particular in the context of cellular transformation, when our own cells can become transformed to the point that they become cancerous. Now, for many, many years, the fields of cancer biology and infectious disease have been separate entities. But what I'd like to argue, and I think many are recognizing more and more, is that the lessons that we learn from the study of cancer can explain what's going on with infection, and the lessons that we learn from infection can help us better understand cancer. The reason for this is because whether you're talking about a cancerous cell or an infectious agent, the immune system has the same problem. You have an entity within your body that can multiply, that can spread, and if not controlled by the weapons of your immune system, can result in the demise of the host. And what we've now learned over many decades is that the ways in which our immune system recognizes infection and cancer, and the ways that our immune system acts to eliminate infection and cancer are very much symmetrical. And this statement supports the idea that a dual focus research area where we try to learn what we can from cancer to explain infection, and we try to learn from infection to teach us about cancer, is a fruitful area of research. And this is really where my lab is focused on, trying to take both of these types of threats to the host and learn as much as we can. These question marks in the center of your slide are really what my lab is primarily focused on a day-to-day level. What does it mean to recognize non-self? What does it mean to recognize dysregulated self? And how do we generate actionable information to save and keep ourselves healthy? So any point on this discussion of self, non-self discrimination has to focus on these cells right here, the dendritic cells, because dendritic cells represent the apex regulators of our immune system. These cells either directly or indirectly control the actions of virtually all other immune cells in our bodies. And for this reason, there's been tremendous interest in understanding how the power of a dendritic cell to control adaptive immunity and local inflammation can be harnessed for therapeutic gain. And of course, at the center of dendritic cells is the idea of innate immunity. And so in order to think about this the way that, or to discuss this the way that we've been thinking about this in the lab, we need to go back in time for a bit. Let's go back to 1989, where the late and great Charles Janeway Jr. proposed this dirty little secret of immunology. So for the students on the call, Back in the 1980s and before, there was a very heavy emphasis on identifying antigens as a means of generating vaccination and protective immunity. But you always needed to mix with these antigens, these chemicals known as adjuvants, that were capable of eliciting an immune response to the antigen that you worked so hard to identify. But this was largely discussed from a technical perspective. It was discussed that, if you want to generate an adaptive immune response, just pull that adjuvant off the shelf. It's simply an additive. But what Janeway realized was that within this additive, within these adjuvants, is immunology itself, meaning that these additives are biologically interesting entities. The general belief in the field in the 1980s were that antigens were sufficient to stimulate adaptive immunity, but the dirty little secret that Janeway highlighted was that adjuvants were needed for antigenicity. By recognizing this dirty little secret, we unveiled the revolution of the modern field of innate immunity, with the new outcome being what we teach in textbooks, what we teach in classes these days, and that is the concept of innate immune control of adaptive immunity, the centerpiece of which, cellularly, are the dendritic cells. Now, I'd like to propose that this was not the only dirty little secret that we need to discuss. If we fast forward to today's world, there's a new dirty little secret of innate immunity. And that new dirty little secret can be highlighted with this concept here, the general belief that necessity equals sufficiency. What do I mean by this? These days, there's a general belief in the field, which has been therapeutically invested in to the level of billions of dollars, that cellular activities that are necessary for immunity are sufficient to stimulate immunity, meaning that antigens that are necessary, CD4 T co-stimulation that is necessary, IL-12, IL-23, interferon, if you eliminate any of these molecules, you can reveal them as necessary to stimulate immunity. And because of that, there has been a significant biotechnology, academic, and pharmaceutical investment in generating adjuvants that stimulate these single events that are necessary. Agonistic CD4 T antibodies, recombinant interleukin-12, antigens in the form of the COVID-19 vaccines and the influenza virus vaccine Afluria, non-adjuvanted antigens. Of course, CD4 T is necessary. Of course, IL-12 is necessary and antigens are necessary, but that does not mean that they are sufficient. I think about this in the same way I think of an automobile. A steering wheel is necessary for me to drive to work every day, but a steering wheel inside of a box does not make a car. There are many ways to break the immune system. You could break the immune system by knocking out IL-12 or IL-23 or any other cytokine. But to turn on a productive response, you do not need to think about silver bullets that are sufficient to activate immunity. You have to think about networks. And that's what dendritic cells really provide us. Dendritic cells are the orchestrators of all of these necessary signals so that you can have multiple signals that ultimately confer protection. These days, we believe that there are at least 5 dendritic cell activities that are important for durable immunity. And when I say durable immunity, I mean lifelong memory T cell generation. These activities include the following: antigen presentation activity, co-stimulatory molecules, effector cytokines such as interleukin-12 or interferon production. These are the big 3 pathways that are typically discussed in immunology classes around the world. But we know that we also need these dendritic cells to move in high numbers to the lymph node where naive T cells abound. If dendritic cells don't move in high numbers to the lymph node, then they are presenting antigen in the periphery. They're not presenting antigen to naive T cells, and therefore you're not going to generate effective T cell responses. And we also know through more recent work, in part from my lab and in part from many, many other labs around the world, that there are specific cytokines that dendritic cells produce that are not necessarily important for effector T cell generation, but rather for memory. A principal example of this are members of the interleukin-1 family, in particular IL-1 beta. IL-1 beta can be considered, as it was once named, a lymphocyte activating factor. We now know that IL-1 beta is absolutely critical for memory T cell generation and for memory T cell reactivation, meaning that if you have a dendritic cell that does not provide IL-1, then you can generate a lot of effector T cells, but they do not sustain themselves durably, and you lose immune memory. Now, these 5 signals, when combined together, are what we now think are important to go from short-term innate immune responses and short-term T cell responses to durable immunity. This point is important to us because when you think about the signal pathways, the signal transduction pathways that are used to activate these responses within dendritic cells, we often think of innate immune pattern recognition receptors, such as the toll-like receptors. But work that has emerged in recent years, in part again from my lab, is that toll-like receptors are not enough. There are other stimuli that are necessary to work together with toll-like receptors in order to elicit all 5 of these activities I just mentioned. So for example, in the center of your screen, you'll see several infection-like signals, lipopolysaccharides from bacteria or RNA-like chemicals such as RA48. These are toll-like receptor agonists, well established. They'll activate select tolls to drive the induction of 3 of the 5 3 of the 5 activities of a dendritic cell I mentioned. They're very good at driving antigen presentation, co-stimulatory molecule expression, and effector skewing cytokines such as IL-12. And the result of a TLR-stimulated dendritic cell is this term that we all use in the field called active. We say that a dendritic cell goes from being inactive to active. But toll-like receptors are not effective at inducing migration. They are poor inducers of dendritic cell migration, and they are poor inducers of IL-1. However, if a dendritic cell is exposed to an infection signal, such as a toll-like receptor ligand, and distinct chemicals that are indicative of tissue injury, which we call hyperactivators, now what happens is the dendritic cell sees an infection signal and a tissue injury signal. It's the combination of these 2 signals that allow a dendritic cell to add activities. When dendritic cells encounter these injury signals or these hyperactivators, which I'll describe more in a moment, you get an increased migratory activity to the draining lymph node, allowing for a higher probability of dendritic cells encountering their naive T cell counterparts. And you unleash the weapons of the NLRP3 inflammasome within a living dendritic cell, such that you can provide this memory T cell skewing cytokine, IL-1 beta. Now, because we are adding migration and IL-1 to the repertoire of immune activities elicited by an active dendritic cell, we say that these cells are not merely active, but rather they are hyperactive. And because of that, the chemicals that we use that hyperactivate these cells, we call hyperactivators. One of which is this chemical here, which I'll now describe, PGPC. So what is PGPC? PGPC is an oxidized lipid that's present and released from all of our cells in our bodies. Anytime that cells or tissues in our bodies die, there will be a necessary release of oxidation products, if for no other reason from damaged mitochondria, which will always depolarize when a cell dies. Now, a damaged cell will release many factors, some of which will be inert, some of which will be immunosuppressive, and some of which will be inflammatory. Within this latter category is this oxidized phosphatidylcholine derivative shown here, known as PGPC. PGPC is released from all of our cells every single time that they die. And in addition, PGPC is chemically synthesizable. You can buy it off the shelf. And if used in proper form, it becomes a very potent mediator of dendritic cell hyperactivation, meaning that when cells are encountering co-ligands, which is an indicator of infection, and simultaneously PGPC, which is an indicator of tissue injury, the combination of infection plus injury equals a dendritic cell that has all 5 of the activities I described to you. Now, in the next several slides, I'm going to show you some evidence that supports this thesis and then explain to you how we've been using these tools as a means to best understand the ways to elicit protective immunity. So first, let's talk about what these cells look like. So if we look at naive bone marrow-derived dendritic cells from a mouse, these cells have been stained with blue for their nuclei and green for their plasma membrane. These cells look like typical dendritic cells. They are amoeboid-like cells, a little bit like a pancake. If you stimulate these cells with the toll ligand LPS, they don't look much different. They don't look very dendritic, but this is typically what we see when we examine LPS-stimulated DCs. If you treat cells with LPS plus an adjuvant known as aluminum hydroxide or alum, the most common adjuvant used in the United States today, what you can see is that these cells will round up and they will float away. This has been known for many years. These cells will undergo pyroptosis. But if we treat these cells with LPS and not alum, but rather a distinct agent, this hyperactivated known as PGPC, you can see these cells with the nucleus again in blue are extremely elongated, very much dendritic, and exactly what you would imagine when we draw cartoons of what a dendritic cell looks like. Very distinct morphology. And this distinct morphology raised the question of what's going on inside of these cells. So to address this, we performed RNA sequencing analysis of CDC1s or type 1 dendritic cells elicited after flat-3 ligand bone marrow differentiation cultures. What you can see is when we compare no stimulation to LPS stimulation to all the others, you can see the major distinction in terms of genes that are highly and selectively upregulated by toll ligands plus PGPC is this module of transcriptional responses, this module of mRNAs that are known as a cell migration module. And this module made a lot of sense because when you look at the morphology of these cells, they look like they are starting to crawl around. And so we decided to ask, what is the difference in migratory capacity between toll-stimulated activated cells versus hyperactivated cells? For these experiments, what we did is we took dendritic cells ex vivo and we stimulated them with either nothing, with LPS plus the hyperactivated PGPC, or with LPS plus this other adjuvant, alum, that I described to you a few minutes ago. We took these stimulated cells and then we injected them into the skin of a mouse, and 24 hours later asked how many of those dendritic cells migrated to the skin draining lymph node. And what you can see is that we can reprove the statement that I made earlier, that toll ligands are poor inducers of dendritic cell migration. We get very little dendritic cell migration. You get even less with alum treatment because these cells are effectively dead. But you can see when we treat these cells with LPS plus PGPC, we get about 30 times more dendritic cells migrating to the draining lymph node, meaning that we have enhanced the migratory capacity of these cells. So now let's shift to the question of IL-1. IL-1 production is typically mediated by the activities of inflammasomes because IL-1 needs to be cleaved by caspase-1 within an inflammasome. And what we found through our studies is that there are two different flavors of inflammasome behaviors within cells. On the left side of your screen is the most common behavior of an inflammasome, shown here in green. And that is that once an inflammasome is assembled, the cells will kill themselves through an inflammatory process known as pyroptosis. We can see that these cells in this image are very much dead because if we watch a movie of these cells and ask them to phagocytose these red-colored yeast particles that are floating by, you can see that this corpse, which we had to pseudo-color in blue in order to see at all, is completely and absolutely inert. These cells have been stimulated with LPS and alum, the adjuvant that's used in the clinic today. Very good for inducing inflammation, very poor for inducing adaptive immunity because a dead dendritic cell is not an effective antigen-presenting cell. But now let's shift to the right side of your slide and stimulate these cells with LPS plus PGPC, which also weaponizes the inflammasome. Here you can see very nice inflammasome speck right in the center of the cell, but these cells are very much alive and are gobbling up through the process of phagocytosis these red-colored yeast particles. And so what that means then is that when you use these hyperactivators, you are adding to the repertoire of the cytokines that can be produced by an active living migratory cell, IL-1. And since IL-1 is important for adaptive immunity, there's a lot of questions that come up. Which of these states of a dendritic cell is best for protection? So we can use this heat map here as a readout or a gauge to teach ourselves what we've learned through these exercises. Naive dendritic cells are very good at collecting antigens, but not very good at anything else. A toll activated dendritic cell are very good at some of these activities, but quite poor at the others. A pyroptotic cell, such as those elicited by alum, are really good at producing IL-1 and really good at dying, but nothing much else. But a hyperactive cell activates all 5 of these important activities that I described to you. And for that reason, we can ask, which dendritic cell state generates the most robust T cells in the context of anti-tumor immunity? So we decided to address this in a very simple way. We're going to take antigens from tumors, we're going to mix them with adjuvants, and we're going to inject that into a mouse and ask if that mouse is protected from cancer. A familiar regimen that we use for the next several slides is as follows. We implant these mice with the tumor. We've used 5 different tumor models. I'm showing you one here. The B16 melanoma model that encodes the ova-albumin antigen. We give these mice cancer for about a week until the tumors are visual and measurable. And then after the tumors have formed, we immunize these mice with adjuvants of interest such as LPS or PGPC, or both, in the presence of an antigen source. And for these experiments, we're using as an antigen source WTL, whole tumor lysates from the same cancer. So we just grind up the cancers, mix them with our adjuvants, and inject them a few times into the mouse and ask what happens. And this is what we found. So if you take a mouse and you do not give them any therapy whatsoever, in black, you see these mice will sadly all die within about a month. If you mix these whole tumor lysate antigens with LPS plus alum, you get very little protective immunity in green. But if you mix these antigens with a distinct adjuvant, LPS plus the hyperactivator, you get a very nice long-term protective response. Now, this result was important for 2 reasons. One of which is because it demonstrates the value of these hyperactivators, which we had previously only been characterizing in vitro in an in vivo model of protective immunity. But secondly, it illustrates the importance of considering the dendritic cell stimulus. The red lines and the green lines were all immunized with exactly the same antigen. The only difference in this experiment is the adjuvant that we used, yet the outcome is dramatically different. So we think about this because, as I mentioned, there has been tremendous investment in tumor antigen and infectious disease antigen identification. That business is a worldwide business. It's very successful. And if you do not put as much investment in the type of the adjuvant that you use, you will not extract the value from the antigen that you identified. The best antigen with the wrong adjuvant gives you nothing. The best antigen with the best adjuvant gives you something very much desired. So we can play games in this same exact module where we can manipulate the genotypes of the mice. So we can get into how we do this earlier, but this is through standard genetic manipulations of these mice so that we could generate mice that have either wild-type dendritic cells, inflammasome deficient dendritic cells, either through knocking out NLRP3 or knocking out caspases 1 and 11, or dendritic cells that lack the ability to migrate from the skin to the draining lymph node by knocking out CCR7, this critical chemokine receptor. All of these mice are only defective for these genes in DCs. Every other cell in the mouse is wild type. And if we take these mice and we give them the same immunization regimen that I described to you, you can see only mice that have wild type dendritic cells can protect themselves from cancer. If they lack the ability to migrate and if they lack inflammasomes, you have a deficiency in protective immunity. This type of evidence actually was, when we published it a couple years ago, the first genetic evidence that inflammasomes, specifically within dendritic cells, was important for anti-tumor immunity. Why are we getting such durable responses? We think the reason is as follows. So if we look within the mouse 200 days after the mouse tumor was eliminated, and we look where the tumor once was, you can see that there's a very high percentage of CD8 positive T resident memory cells, which are classically associated with strong anti-tumor immunity. These CD8 positive TRMs are not present at the site of immunization. They are present at the site of where the tumor was. And this allows us to build a model whereby you can immunize a mouse in a distal part of the body. You can weaponize your T cells in the draining lymph node because of all the dendritic cells that are flowing into that part of the body. The T cells exit the lymph node, hunt down the tumor, eradicate the tumor, and then guard the site where the tumor once was to prevent it from recurring. We think these CD8 TRMs are important because when we pull these TRMs or circulating memory cells out of what we call survivor mice, so mice that survived the tumor 200 days ago, and we ask these T cells, can they display cytolytic activity to B16 tumors in vitro or B16 ova tumors in vitro? The answer is yes. What we're measuring is the ability of these T cells to lyse the correct tumors, but notably, they cannot lyse different tumors. And moreover, if we take these cells and we transfer them into a naive mouse that has never seen anything else, you can see if we transfer in the TRM cells alone or the circulating memory cells alone, but much more impressively, if we combine the circulating memories and this resident memories into one injection, and we inject these mice with these T cells from a survivor, we get complete protective immunity in an otherwise naive mouse, meaning that these TRMs are sufficient to convert protection to these tumors in vivo, and they display lytic activity toward those tumors in vitro. How long do these immunizations last? Everything that I described to you was short-term experiments where we give mice cancer and then about a week later we immunize these mice. What happens if we immunize the mice first and ask how long can the immunization confer protection? In order to address this, we focused on the ova-albumin antigen itself. We immunized mice at 8 weeks of age, and then we waited over a year for these mice to progress. After over a year, we gave these mice a boost once of antigen alone and then challenged these mice with tumors that contained this antigen, B16 ova mice. And what you can see on your right is that if we immunize these mice with LPS plus PGPC as the adjuvant and ova as the antigen, even a year later, we get 100% protective immunity. But if we use LPS or LPS plus PGPC in an NLRP3 knockout, we get very minimal, if any, protective immunity that lasts over a year. If we use LPS plus alum, which I've already shown you is a poor inducer of protection, we get almost no protection whatsoever. What this allows us to do is start talking about questions of immune durability. It also raises the question of what happens in an old mouse or an elderly mouse. And I bring this up because I think we can all agree that while cancers occur at all ages of human existence, it is principally and primarily a disease of the elderly. The elderly are one of the largest risk cases for cancer progression. There's been a lot of discussion of this over the years. There have been many defects in immunity that have been documented in the elderly. I'm showing you just a few here. Primary immune responses are weaker as we get older, very sadly. Memory responses are lower, recall responses are weaker, and individuals are more susceptible to cancers as we get older. And so for the next several slides, I'm going to present to you a recent publication that we had where we examined anti-tumor responses in old or young mice and asked, can we start learning something about what's wrong in the elderly? And the reason why I bring that up is because while there have been tons of literature on what is wrong, there are very few instances of correcting those problems. Can we use the power of the innate immune system to correct immune defects in the elderly? We first began by looking in mice of various ages, 8 weeks, 20 weeks, 40 weeks, 68 weeks, and 90 weeks of age, and asked what is the repertoire of CD8 and CD4 T cells present within these mice. What you can see is that as mice age, they start dramatically contracting the frequency of CD8 T cells that are present within the body. And perhaps more alarmingly, as mice age, the percentage of naive T cells drops very, very low. If you have less repertoire of CD8s and you have a lower repertoire of naive T cells, then of course you're going to have problems with generating new immune responses as we get older. And this is a problem for infectious disease vaccinations in the elderly and for perhaps anti-tumor immunity. So we wondered what happens with mice as they start getting older. First, we wanted to determine if there's any problem in dendritic cells in this context. What I showed you here are problems with the T cells. But what we see in dendritic cells is that not only is it the T cell compartment that's problematic, the dendritic cell compartment is also problematic. If we look on the left side of your slide at 8-week-old dendritic cells or 68-week-old dendritic cells, you can see that while LPS-stimulated dendritic cells will migrate from the skin to the draining lymph node at a pretty weak rate, you can still detect it. But if you look at elderly dendritic cells in black, you have almost no migratory activity. What that means is that you have poor antigen-presenting cell activities and you have few naive T cell activities in the elderly. But what you can also see is that when we take elderly dendritic cells and we expose them to LPS plus our hyperactivator PGPC, we can correct this defect. We go from minimal migration in the elderly to very strong migration in the elderly. And of course, we're getting very strong migration in young mice, as I showed you previously. All other dendritic cell stimulants we examined were unable to correct these defects in the elderly, only hyperactivators. And what was notable is we can play the same game in monocyte-derived dendritic cells that we isolated from a collection of human donors, all of which was 70 years old or greater. What you can see is that unstimulated dendritic cells, and here this is a transwell migration assay, unstimulated dendritic cells migrate very poorly. If you stimulate cells with LPS, they don't migrate very well at all. If you stimulate elderly dendritic cells with LPS plus PGPC, you get very strong migratory activity, but not with any other migration or any other dendritic cell stimulant. And so what that allows us to conclude then is that we have less CD8 T cells, we have less naive T cells, we have less migratory DCs. What happens to cancer and cancer therapies in elderly mice? We began by first asking what happens with cancer. What I'm showing you here is a set of graphs. We did not give these mice any immunotherapies, we just gave them cancer. They were all implanted with the same tumor, and we've done this with 4 different types of tumors. I'm showing you one here. If we compare 8-week-old mice to 20-week-old mice, the tumors kill mice at about the same rate. 8-week to 40-week-old mice, you can start seeing that the 40-week-old mice are dying a little faster than the 8-week-old mice. 68-week-old mice, the same thing. 92-week-old mice, even faster. So as mice are aging, they're losing the ability to intrinsically control the cancer. There's a defect in cancer protection as we get older. And this, of course, correlates superficially at this moment with the defects in dendritic cells and defects in T cells. So can we move from this correlation analysis to function? That's what we're going to discuss next. We asked, can we treat these mice with checkpoint inhibitors, which are so successful and successfully used in the clinic and in young mice? And what you can see is that with the young mouse, if we give these mice implantable tumors, normally they'll pass away. They'll die within about a month. But if the mouse is young, checkpoint inhibitors based on PD-1 are very effective at protecting these mice. I'm repeating work that's been done by numerous labs around the world here. But what was news to us was when we do the same experiment in an old mouse, you can see that these checkpoint inhibitors are very much ineffective. This was true for PD-1 treatments. It was also true for CTLA-4 treatments, and it was true for PD-1 and CTLA-4 combination treatments. As the mice got older, these treatments lost effectiveness. In contrast, it didn't matter how old the mouse was. If we give these mice an implantable tumor and then we immunize them the way I described to you earlier, using tumor antigens as our antigen source, regardless of the age of the mouse, we got very nice, durable, protective responses. What that means then is that dendritic cell targeting immunotherapies can correct age-associated defects in immunity, whereas the T cell targeting therapies that we've tested cannot. Let's look a bit more closely at the value of PGPC-based immunizations as compared to other ones like I showed you earlier. Only in old mice do we see LPS plus PGPC eliciting this long-term protective response. Any other adjuvant conferred no response, and if we look at the growth of these tumors, you can see that if you use the wrong adjuvant, or let's just say a different adjuvant, you get very robust tumor growth. With LPS plus PGPC, we have most of the mice had tumors that were effectively running along the edge of the x-axis. So protection correlates with tumor restriction. What was notable here is this phenotype that I described to you, where we were losing CD8 T cells. If we're losing CD8 T cells as we get older, that could explain why checkpoint inhibitors are losing efficacy. Because many folks believe that CD8 T cells, and in fact, there's significant evidence to support this, are the targets of checkpoint inhibitor therapies. But the question came up, if we're losing CD8 T cells, how are we still getting protective immunity by dendritic cell hyperactivating vaccines? This raised the question of which T cells are responsible. So in order to address this, we did the same type of cancer experiment I described to you, but here we depleted CD4 T cells or CD8 alpha positive T cells from these mice. And you can see in young mice, as one would expect for all other immunotherapies, CD8 depletions in black, when we use hyperactivated vaccines, totally eliminates protective immunity. CD4 T cell depletions did not impact protective immunity. The opposite was the case in elderly mice. In elderly mice, when we depleted CD4 T cells in black, we lost protection, whereas CD8 T cells were not important and not required for elderly mice to protect themselves. This suggested then that as we age, dendritic cells can best weaponize or best utilize different arms of the adaptive immune system. If we look within the tumor at what's going on here, specifically focusing on the CD4s that are important for protection in the elderly, you can see that within these elderly mice, once we immunize these mice with our adjuvants and with antigens corresponding to those tumors, you can see a decrease in the regulatory immunosuppressive T cells within the tumors, and you can see an increase in the percentage of interferon gamma positive to IL-1 positive ratio, IL-10 positive ratio, meaning that we are inflaming or warming up the T cell repertoire in the tumor microenvironment. This raises the question then of what do these T cells actually look like? So we collaborated with Or Aschenberg and Romnick Xavier at our institution to perform single-cell RNA sequencing analysis of T cells isolated from the lymph nodes of mice that were immunized with different adjuvants and a fixed antigen. I'm going to show you some of this data right here. First, to make sure that the data analysis looked good, you can see that we can very cleanly identify all the major T cell subsets we were interested in. What was notable is that when we look within these T cells, you can see a very distinct state of individual T cell types based on the age of the mouse. So age has a tremendous impact on the transcriptional repertoire of T cells. And if we compare non-immunized mice in light blue to immunized mice in dark blue, you can see that it's not just age that impacts the T cell transcriptome, it is also the immunization. that impacts the T cell transcriptome. So now the question becomes, what's the difference between these T cells in young and elderly mice? I'm going to show you several data sets on this topic. The first, if we look at naive T cell scores based on transcriptional anchors for these labels, you can see, as expected, 8-week-old mice have a lot of naive T cells. Elderly mice have very few naive T cells. This is consistent with the flow cytometry data I showed you previously and consistent with work from others over the years. Also consistent with prior work, you can see that the large signature of T cell exhaustion is present within T cells isolated from old elderly mice versus young mice. If we look at the inflammatory Th1 score of these T cells, you can see that CD4 T cells have a very strong T cell score as far as Th1 signatures go, as you look at old mice, elderly mice versus young mice. But what was most notable in this data set was when we looked at cytolytic activity. This is within the CD4 T cell repertoire, and you can see that the CD4 T cells have a very large component of what is classically considered a CTL, or a cytolytic signature, but only in elderly mice, not in young mice. This data suggested transcriptionally that dendritic cell hyperactivators are capable of inducing cytolytic activity in a CD4 T cell. In order to address this, we decided to perform the following experiments. We immunized mice at 8 weeks age or 92 weeks of age and pulled from these mice CD8 T cells in this slide or CD4 T cells in the next slide, and asked, can these T cells display cytolytic activity towards B16 ova cells ex vivo? What you can see, if we're measuring degranulation of the T cells by the surface exposure of CD107 alpha, you can see that, not surprisingly, young mice in red here that were immunized with OVA plus LPS plus this hyperactivated PGPC, they're capable of degranulating when exposed to OVA-expressing cancer cells, as you can see in red. You get a little bit of degranulation in elderly mice as well. But what's notable is that this little bit of degranulation did not correlate with cytolytic activity. If now, rather than looking at degranulation, we look at lysis of these cells by measuring the absolute number of cancer cells in the well after co-culture, or measuring the production of lactate dehydrogenase, which is a readout for cell rupture in the well, you can see that young CD8 T cells are very good at killing cancer cells, and they're very good at lysing cancer cells. Elderly CD8 T cells are very poor at killing these cells. In fact, they're no better than naive T cells that were pulled out of an unimmunized mouse, and they can't induce lysis. So this data suggests that as we age, or at least as mice age, CD8 T cells are not only less in abundance, those that remain are poorly cytolytic. Now let's shift the discussion to CD4 T cells, and what you can see is exactly the opposite. In young mice, CD4 T cells display no evidence of degranulation. They display no evidence of lytic activity in either of the assays we used. But in elderly mice that were immunized with hyperactivators, and only hyperactivators, you can see degranulation from these CD4s, you lose the cancer cells, and you lyse these cancer cells, meaning that just as our transcriptional analysis predicted, elderly CD4 T cells can be weaponized to display cytolytic activity, functionally and transcriptionally. And what was notable about this data set is it may explain the following slide right here. If we take CD4 T cells out of an immunized elderly mouse, and we transfer these CD4 T cells into a naive mouse, we can protect these mice from implantable tumors. But if you take CD8 T cells, B cells, or even monocytes out of an immunized elderly mouse, and you transfer them into a naive mouse, you get minimal protection whatsoever. In this transfer experiment, we depleted from the recipients T cells, meaning that the CD4 T cells that are transferred are not getting help in terms of protective immunity from any other T cell that's present within the recipient mouse. And this data supports the idea then that there can be a way to best utilize CD4s in the absence of CD8 T cells in the elderly. So let's take this data set and consider it in the context of what's been done therapeutically. What I'd like to close with is this discussion. Prior strategies of generating cancer vaccines have, as we all know, been unfortunately disappointing. There's been tremendous interest for over 40 years in trying to take advantage of the fact that we can generate infectious disease vaccines. Why can't we generate cancer vaccines? And most of the focus in this area has been on antigen identification, neoantigen identification. There's been mountains that have been moved in this space, and my hat is off to the scientists who have done this marvelous, comprehensive, and rapidly accelerating work. But one of the challenges with the field is that typically what we as a community do is take our antigens that we've worked so hard to identify and mix them with toll ligands, or mix them with alum. As I mentioned, the most common FDA-approved adjuvant on the market today. Now, as a basic scientist, I would tell you that toll ligands and alum do not elicit all of the 5 dendritic cell activities that are so necessary for durable protective immunity. And for that reason, if I can go back 30, 40 years, or even 10 years ago and spoken to myself working in that area, I would say this approach is likely to yield a disappointing clinical outcome because you're not going to be generating effective, durable memory T cells. And this is exactly what we've seen in the cancer vaccine space. Disappointment after disappointment. But what I've shown you today is that there may be a solution for this problem. Perhaps we don't need to have another innovation in how antigens are identified. Perhaps what we need to do is take the antigens that we meaning the world has so, worked so hard to identify, but use them with the best adjuvants. Using hyperactivated dendritic cells, you get the value of an inflammasome to provide IL-1 that drives memory T cell activation and reactivation. You get a dendritic cell that migrates to a much higher rate to the lymph node, therefore allowing you to utilize the antigen presenting capacity of those cells at a much higher frequency. And at least in these preclinical models, the outcome is what I would like to propose: lifelong immunity, at least in an animal. And so to summarize, what we believe is that hyperactive dendritic cells provide all of the classic signals that other stimulated dendritic cells do: MHC, co-stimulatory molecules, and effector cytokines. But the reason why they're special is because they can induce hypermigratory activity and IL-1 from a living cell. And this flexibility of the best weaponized dendritic cell allows you then to be almost, if I'm going to be ambitious here, age agnostic. In a young mouse, these dendritic cells will probably best utilize CD8 T cells to eradicate tumors. But in an elderly mouse, the same exact immunization regimen will use the T cells that are there, and that just happens to be CD4s. There's a lot of questions that come up from this work. We still have much effort to explore this in the context of protective immunity of different cancers, infectious disease, different ages. Many questions are on the table. But the hope is, with this presentation, that we can continue to emphasize the value of different dendritic cell activation states in order to elicit the types of protective immunity we need at all stages of life. And so with that, I'd just like to thank the principal scientist who did the work on this project, Danya Savaki, a spectacularly talented scientist who really began all of our cancer work in my lab. She's masterful with hands, a brilliant mind. And of course, I want to thank the folks on the right, other members of my lab on the left. And I want to thank you all, most importantly, for your attention. Thank you all, and have a good day.
Well, thank you, John, for the excellent and very passionate talk. As usual, I know we would all like to ask many questions now, but I have to refer you to Twitter or to X for asking those questions, and I hope that there will be many questions to John. But normally I'm not supposed to ask questions, but this time I would also like to ask you a question. It's very fascinating to see a different response in the old and the young mice. I'm wondering, have you done any work with OT1 T cells, transfer them into the old mice, and
Could you,
so, what was
if
their
we've done
response?
anything with what?
OT1 T cells, OT1 TCR transgenic T cells,
Ah,
and just
yeah.
see
So
if they respond like young, or is the environment of the old mouse suppressive, and then you see sort of the same response even though you transferred OT1 T cells, maybe from a
it's a
young
super
mouse.
question. Yeah. So we have transferred ova-specific endogenous antigen-specific T cell response. We have not transferred OT1 or OT2 cells. But I should highlight a paper that recently was published by Arlene Sharp at our institution, who really did a very nice job of exploring some of these questions in an analogous way. And the work of our lab, of Arlene's lab, and Deb Sen's lab at Mass General Hospital, amazingly also in Boston, on kind of problems and success stories in the area of age-associated T cell responses, is something that is attractive. So that's a long way of saying that there's a lot to do. There's a lot that's ongoing, and I'm really excited to see how this field progresses.
Okay, cool. Thank you very much again. With this, I'd like to close today's meeting. Thanking you again for being our presenter today. And I would also like to announce that next week our speaker is going to be Cornelius Murrer from UCSD. With that, have a great remaining day to everybody. Thanks, John, and I hope to see you again next week.
Right.
Bye-bye.
Very good. Thank you all.
Bye-bye.